All right, welcome to another episode of the Bioinformatics Lab podcast. I'm Kevin Libwitt with Andrew Page. We're from Theogen, and today we're going to be talking about data sharing and the conversation regarding metadata or contextualized data and the difficulties and lack of standards, maybe is a way we can describe it around this area. I think we saw this at the forefront of a lot of conversations, especially on everyone's mind during the COVID-19 pandemic, when we were all trying to share information and contextualize what's actually happening with the information being shared. It wasn't exactly obvious to even understand even just the minimal contextual information of where this was from, the data was collected, and primer sequence that was utilized to generate the data. So there's been a huge amount of efforts in working groups that I know you're a part of in terms of the phage universe to address this problem. But I'm guessing this is something that you've seen throughout your career, even before the pandemic. If you want to maybe start us in that conversation.
Yeah, I guess this has been sparked by a paper from phage, from the Data Standards Working Group or Data Structures Working Group. And yeah, what always seems to happen is people don't share their data, or they share it with their, no metadata, or the wrong type of metadata, or incompatible metadata. And then it's not as useful as it should be. Ideally, if you get an outbreak somewhere, you should be able to go into public archives and say, right, people have seen this in 10 other countries. It's in nuts, or it's in, this type of environment. And then you can go from there and you can actually make quick, rapid decisions. But often people don't put up that information. Even the most minimal of minimal information is missing. So what I've seen, and I'm partially at fault, I'll put my hand up to that. Like when I was at Sanger, just the way the systems worked, we would accession samples as they came physically into the building. We wouldn't necessarily have the metadata to go with those, that would come later or might have to be pulled together. So the samples would often or usually be sequenced before the metadata arrived. And to get around that, we'd create a biosample, which is like the metadata placeholder in INSDC for a sample. And that would just have basically the species that we were told it was. Doesn't mean it is the actual species of the sequencing. So just beware of that. And then basically some kind of identifier, and that was it. That was the absolute minimum that had to go in, which is not useful at all. And then the idea was it would be sequenced, and then, people would update the metadata later, but often that didn't happen. I know there's a bit of a backfilling operation later, but, sometimes this metadata would be stuck in, supplementary materials for papers or in PDFs or things that aren't very useful to people. And so that's kind of a historic legacy there. And people are trying to correct that to actually make stuff useful. So there's this pathogen data object model from phage, and they're trying to address this, just to do a little bit better with data sharing and metadata.
And I think what you described has traditionally been the common model, where there's like a single institution that may be receiving samples from a number of different places. And it might be just a specimen and really minimal information about what kind of specimen you're receiving, and not that other contextual information. So they might not even have access to the information to share to INSDC or whatever international data repository. And also one thing I would say that we've noticed is that a lot of those laboratories are maybe in some way academic. Whereas now, especially during the pandemic, so many more public health laboratories were equipped with sequencing instrumentation and bioinformatics capabilities, and they were much closer to the contextual information. And they needed that contextual information to make decisions and understand what was happening with SARS-CoV-2. So it was the timing to speak to that audience what was prime during the COVID-19 pandemic there.
But also public health labs don't necessarily have the ability to share data.
Yeah.
So like a lot of UK data, if you have a look at it, it'll look like one place in London called Colindale is like a hotspot of all disease. But that's because that's where the lab is based.
Yeah.
For UKHSA or Public Health England, as it used to be called. And so that's just, within their framework, they just cannot release the data unless you come to them and say, these are the reasons. And then they'll share more, or they'll share it with other public health bodies quite easily, say through Genome Tracker or whatever, through private means. But it's a really interesting thing. You can't necessarily trust all the data you get. And also, they're very important fields that are often missing. Like, is this person a traveler or not? that's a key piece of information because,
Mm.
if they've come from somewhere else in the world, it makes a big difference versus someone who picks up a disease, or picks up an infection, in the local community. So, you might be saying, oh, well, is this a multi-country outbreak or is it just you've got a few travelers who've gone to other countries? Big difference, and it's very important.
Yeah, that's absolutely right. During the pandemic as well, there were so many lessons learned there, but it was also a lot of conversation of the restrictions of even the geographical information that you were able to share. You said often it was either country or state. In a lot of previous frameworks, it would be that information was maybe delayed release to some extent. So like you couldn't track it back to the individual or the patient. So that was the whole schema of protection is you don't want to release information, obviously, that was personally identifiable or personal health or constitute as personal health information. So there was a lot of restrictions on that. But then, during the pandemic, it's not like you'd be able to identify the person it was when there was hundreds or thousands of cases at a time. So there was more discussion of what level of or what resolution of information could be shared. And I think that's what's helpful about having something like this proposed data object model, in that it proposes what are the minimum pieces of information that are really relevant to public health, that make this public health or make this information useful for the public health community when you're sharing this information. And this is something that this working group, your working group with Emma, led by Emma Griffith in Finn over there, the Canadian public health bioinformatics scientist there, that they put out with SARS-CoV-2 in mind, and then have now since adopted it to really all pathogens or infectious diseases of public health concern.
And what really is good about this particular paper is that it has representatives from the different major INSDC archives. So you have the Europeans, Americans, and Japanese on there, and you have representatives from all the very big producers of data like the FDA and CDC. So it's quite nice. You've got effectively buy-in with all these authors on it, and it means that it's actually more likely to be used, implemented. And it's not just yet another standard that comes out and people forget. It is actually something that is practical and useful, which a lot of academia isn't, because you don't get funding for the, practical necessarily, because that's routine and mundane, but it's really important.
Yeah, again, something that has been with public health, having that closer relationship with academia over the past couple of years, you're seeing these pragmatic efforts being addressed and prioritized, especially again with public health. If it was only an academic exercise, it'd be driven off of curiosity. And okay, we could maybe tag people, really suggest heavily, hey, this information would be helpful. But in the case of public health lab, you have public health stakeholders that are saying, no, this is the information that needs to be shared. Otherwise, we can't look at trends. We can't understand the geographical patterns. We can't even look at a high level of surveillance of how the disease is spreading. So I think you're right. This emphasizes the pragmatism, a step forward in standardizing the field. So yeah, huge congrats. And again, this paper in the data structures group. So how do you see the impact of this now? And what's the next step forward now that this is proposed as the pathogen data object model? How do you see adoption maybe
Well, I
being
guess if the
distributed?
major producers of data adopt it and then the archives allow for it, then you have that, perfect way of bringing everyone else along, because you need that critical mass and you need people to, take that first step. And it's hard being the first people to do things, but it's required. And then, obviously you'll tease out other issues that pop up along the way. I think probably one of the most important things is actually around ontologies and actually having a common
Yeah.
language that's controlled. Because, around the world, we don't all speak English. Or even if we do speak English, we have different words to mean the same thing. Like you'd say tap, or we'd say tap, you'd say faucet. And, if a sample is collected from that and you don't understand the differences in language, well, then, you can come to incorrect conclusions or even nuts, different people will classify, say, nuts in different ways. Some people would say, well, peanuts are not, or ground nuts are totally different to, say, hazelnuts and things. And that is an important distinction as well, particularly around food. And so it is very important to have this kind of common language that anyone can tap into and say, definitively, absolutely, we understand what this is, categorize it correctly, and use it.
Can you speak on that a little bit? The conversation of ontologies, because that's something I like had to learn about coming into the field of bioinformatics. Otherwise, I didn't realize why it was a challenge describing something as chicken, poultry, farm, animal. So yeah, that's a big part of this as well. Do you mind maybe explaining ontologies and its relevance to public health and bioinformatics?
Yeah, it is. It's very, very niche. Some people get really excited about it, but it is super important because if you think about chicken, right? It did the person eat, say, cooked chicken in a restaurant and get ill, or was it, you know, something they cooked at home, because they bought a full thing and made it themselves, or is it, the chicken on the farm and the farmer got infected? So whatever point in the life cycle it is, it's important to know because you can have different responses and different measures can be taken, different conclusions can be drawn. If it is, say, just someone, maybe handling chicken poorly at home, that's not going to cause many problems, bar the household. But if it's within a factory producing products for, millions of people, then that could be a huge deal. And so it is very important to understand the nuances and to get that across to other people. Additionally, we live in a very international, globalized world, and so products and ingredients can come from all over the world. It can be difficult to track things down. So, for example, in the UK or in Europe, we had an outbreak in chocolate. So these little kind of Kinder eggs were causing issues that children eat. And of course, you don't want children to get sick. And tracing it back, they identified, okay, it's a factory in Belgium, and tracing it back further, it wasn't just that factory; it was another factory that supplied product to multiple factories, and it just happened to get into that one and cause problems, and then in those products, and of course, chocolate has a long shelf life, so it goes everywhere, and that went all over Europe, and so having being able to trace that back and actually definitively put into a database and say, this is the thing that is making people sick is really important.
And exactly, that's where these ontologies come into play of these systems of formalized language that are computer readable, really, right? Otherwise, you'd have to have a human interpreting. Oh, when they say poultry, I think in this area of the world they mean a backyard chicken. Whereas maybe this guy, I know, oh, that was Andrew. He put poultry, but what he really means is chicken dinner and specifically breast meat. So when you have these ontologies, these controlled formal categories of a strong language, you can make these connections. You can do some computational analysis to understand what we're all talking about. So that is a huge effort. It's a really difficult thing to get done and standardized and adopted. I think the first time I was ever introduced to the concept of ontologies and its importance was with antimicrobial resistance and the card database. That's where I first saw. I was like, oh, this is the beauty of an ontology. Because before that, it was like I was tracking down gene synonyms and trying to figure out what is the function, what is the protein family, and otherwise. But seeing an ontology layout where I can click a gene and see how it's related to this whole network of terms, I was like, oh, this is an ontology. And now you're seeing that kind of pushed also with the metadata here. Because I think even now to this day, if I go into NCBI pathogen detection, you're going to see the lack of that structure, and you're gonna almost thirst for it, right? You're gonna see like leafy greens, lettuce, salad, or something like that, all trying to tie it back to the same source. But they may all you don't know if they mean the same thing or something different without an ontology and
Yeah.
without this kind of language. So yeah, I think you're seeing the utility in that already.
And also then there's other things like just defining terms, say dates. Dates are a huge thing. It's very important to know, did something happen yesterday or a year ago or 10 years ago? And between obviously Europe and America, we write dates differently. You guys put the month first, I think, and which is bonkers and crazy. So you can very easily misinterpret things because they look the you can dates that look the same. But it is important, when you start drilling down, when you get a date and a sample, what does that mean? Is that the data was received in the sequencing lab, the data was released, the data arrived in maybe the original extraction lab, was the data was actually collected on? So there's so much nuance in something that seems so simple, but it is so important. And then other labs will for legal reasons will put in jitter into their dates, because maybe it's something quite rare. They
Yeah.
don't want people to know exactly when it happened, so that can cause confusion as well.
date formats are nuanced and hilarious conundrum for data scientists. I had to look it up exactly the ISO standard because I know for the date format fanatical groups, the ISO 8601, if you're familiar with that, which is the four digit year, 2-digit month, 2-digit day, that has become the default, and that is definitely the one I propose for most people to adopt. But this was a great conversation. Yeah, I think the conversation of standardizing the field, standardizing the types of data that goes in, and even the way in which that information gets deposited to the databases is an amazing step forward. So congrats to you, Ruth, Timmy, and I believe was the lead author in that, and then all the other folks in the data structures working group, again, something led by Emma Griffith, Finn McGuire, through Phage. Another huge step forward. Yeah, any final words on this?
No, I'd say we'll put the paper into the into show notes and have a read of it.
All right, see you all in the next one.